biotechnology. And for further information, as always, you can look at biotechnology and molecular biology, the separate Wikipedia articles. Let's continue further. So biotechnology is the use of cells or living organisms to develop products for humans. It includes tools such as recombinant DNA, which are DNA molecules formed by laboratory methods of genetic recombination, such as molecular cloning, which bring together genetic material from multiple sources, creating sequences that would otherwise not be found in a genome. Other tools include the use of genomic libraries, DNA microarrays, expression vectors, synthetic genomics, and CRISPR gene editing. So very interesting stuff here. So let's look at this figure right here. So construction of recombinant DNA, in which a foreign DNA fragment is inserted into a plasmid vector. So if we have this host plasmid, remember just a circular DNA, separate from the main chromosomal setup, and we'll just go do a search right here to refresh. So if we go back here, a plasmid is a small extra chromosomal DNA molecule within a cell that is physically separated from chromosomal DNA and can replicate independently. In nature, plasmids often carry genes that benefit the survival of the organism and confer selective advantage, such as antibiotic resistance and so on. So yeah, here's an example just outside of this, the main DNA in the chromosomes. So let's continue further. So search plasmid and go all the way back here, and that is going to be insert into a plasmid vector here. All right, so we have this. So you have this host plasmid, then you have a site of cleavage where you're going to cut it. So you're going to cut it somewhere across here, and then cleavage by restriction endonucleases. These are enzymes. So and then you have these sticky ends there. So you remove these, and you have and these are specified genes that so you want. Yeah, once you cut it off, and now you could insert that in, or you could even cut off even further. But anyway, so all they're doing is cutting off, and then you have specified genes that you want to insert into the genome. So now you're going to anneal. This is called annealing, and then you have recombinant plasmid DNA is you added these to it, and point of attachment and annealing are sticking together. So just note, genetic recombination, also known as genetic reshuffling, is the exchange of genetic material between different organisms, which leads to production of offspring with combinations of traits that differ from those in either parent. Now, in eukaryotes, genetic recombination during meiosis can lead to a novel or new set of genetic information that can be passed on from the parents to the offspring. And most recombination is naturally occurring. Yes, interesting. So now molecular cloning is a set of experimental methods in molecular biology that are used to assemble recombinant DNA molecules to direct their replication within host organisms. The use of the word cloning refers to the fact that the method involves the replication of one molecule to produce a population of cells with identical DNA molecules. Molecular cloning generally uses DNA sequences from two different organisms. The species that is the source of the DNA to be cloned, and the species that will serve as the living host for replication of the recombinant DNA. Molecular cloning methods are central to many contemporary or current or modern areas of modern biology and medicine. In molecular cloning, a vector, so this one is a plasmid vector right there. And so a vector is a DNA molecule used as a vehicle to artificially carry foreign genetic material into another cell. Yeah, so in this case, you're inserting artificial genetic information inside this plasmid, then you could use that as a vector containing genetic material. And you can insert that into another cell. So going further, so annealing in genetics means for complementary sequences or which again are anti-parallel mirrors of each other of single-stranded DNA or RNA to pair by hydrogen bonds to form a double-stranded polynucleotide. So basically, all you're doing is means for when you're dealing with complementary sequences such as DNA or even RNA. Yeah, and then you can pair up the complementary sequence of that. So for example, this one here. So these are a reverse of each other, and you can go and add a complementary setup on this, and then it sticks with hydrogen bonds, and you anneal those together. Yes, very interesting. So now let's continue further. A restriction enzyme or restriction endonuclease endonuclease or restrictase is an enzyme that cleaves or cuts or splits DNA into fragments at or near specific recognition sites within molecules known as restriction sites. So site of cleavage or restriction site right there. So cleavage by restriction endonucleases. And then, yeah, so it's always basically enzymes for everything. All right, let's continue further over here. Now this is a big diagram here. So diagram of molecular cloning using bacteria and plasmids. All right, so let's just zoom in here from the starting point at the top. Zooming in, further center this. Zoom in even further. Let's zoom in even further. So what we have is, number one, we have small circular DNA molecules called plasmids are removed from bacterial cells. These plasmids serve as vectors, molecules which carry genes of interest. So you have bacteria, there's the bacterial chromosome, and then you have the plasmids here. This plasmid includes antibiotic resistance genes, a gene responsible for blue color, well, it's LAZ, and within the LAZ gene, a multiple cloning site, also known as a polylinker, containing various restriction sites. So that's the plasmid right there. So here's the plasmid. You have antibiotic resistance genes, LACZ, or the gene which is blue color. Yeah, so a gene responsible for blue color. Then you have multiple cloning sites and then also they call that MCS. And then there's restriction sites that are the specific recognition sites. So basically, you have the plasmid here. And it has a bunch of other genes on them and some antibiotic resistance genes. So what you can do is, well, let's look at step 2 of this gene cloning. So cell containing gene of interest. Yeah, so this cell has a gene of interest. This one has the vector, which is this plasmid. So DNA containing the gene of interest is also taken from its cell. All right, so you take this, and then here's the gene of interest on this DNA strand there. And there's restriction sites from the starting and end point, and you have a restriction enzyme to link up at that spot. So restriction enzyme recognition sequence is over here that you want. So a restriction enzyme, also called a restriction endonuclease, a nuclease recognizes its specific restriction site, a short sequence of about 4 to 8 base pairs long. Yeah, so a specific one that you want. So this GC, AT, AT, TA, TA, CG. So you want that. Yeah, so you're gonna have you're gonna cut that up, DNA cleavage, and then you're gonna have sticky ends that want to connect with hydrogen bonds. And then there's the restriction enzyme that also cuts up at this specific location that you want to cut up from this and this, and then you're gonna swap them over. So what you can do is, it breaks apart the DNA, leaving overhangs called sticky ends. The restriction enzyme cuts open the circular plasmids. The same enzyme cuts out the gene of interest from its DNA molecule. So you cut out this, and you cut out this. Yeah, so this is the one that at the ends, and this one's you just cut it over, and you're going to insert that inside there. So what you're going to have is, over here, yeah, so you're going to have this, you cut it open, now you have this restriction fragment that you've removed, and then, actually no, the restriction fragment's over here, and then, then you're going to put it over inside this split there. So it splits, and you're going to put that in there. And it's going to stick with hydrogen bonds. All right, the next one here. So hydrogen bonding, and it sticks over to them. So fragment insertion, put it in there and it sticks. And now what you're left with, with this setup. So the sticky ends of the restriction fragments attach to each other via base pairing, forming weak hydrogen bonds. The genes of interest get included in some of the plasmids, forming recombinant plasmids. Other plasmids close right back up, remaining unchanged. Yeah, so other ones that you cut up, these don't end up getting on them. So now you have this close up right here, and you're back to normal with those ones. But some of them have these setups over there. Yeah, so they're weak bonds, and what you're gonna do next is a DNA ligase makes the bond permanent by attaching nucleotides to each other with phosphodiester bonds. And again, this is another enzyme. Yeah, so basically it strengthens that connection there, and then there's those phosphodiester bonds, which is typical in nucleotides. You connect those together. Yeah, so those are the bonds, and then because before we just have the hydrogen bonds, and now you want to connect the actual backbones together. If you have the phosphodiester bonds, which connects the, I believe the sugar and the ribose or the deoxyribose with the phosphate in the backbone. So it's fascinating stuff. So let's continue further. All right, so now let's go to step 7. So this is an interesting one. So the plasmids are mixed with the bacteria. Some of them take up the plasmids in a process called transformation. And note that initially we would take out these plasmids from this bacteria. So you take them out for the ones that have the blue color gene as well. Yeah, so there's a gene responsible for blue color. Take them out, and then so then they won't be blue anymore. And then when you did this recombinant plasmid DNA setup, what happens is so you would cut these up and then put them inside and then glue them back up. And some of these will have it. So the genes of interest get included into some of the plasmids. So the key is some. Some of the plasmids forming recombinant plasmids. Other plasmids close right back up, remaining unchanged. So you have some unchanged. And then what you can do is the plasmids are going to be mixed back in with the bacteria. And some of them take up the plasmids in a process called transformation. So you have transformed bacteria. This one's going to be blue. Yeah, this one's going to be blue. This is the unchanged one. So unchanged plasmid, it's still going to be called transformation. And then the transformed bacteria that are both going to be transformed is the one with over here. This is the one with the new setup right here. And note that this one's not blue. And then these other ones are untransformed bacteria that don't have the plasmids. These are also not blue. So in this way, you can tell which ones had the original plasmid setup, but now you have a scenario where you have one that's not blue and another one that's not blue. This one's untransformed with no plasmids, and this one is transformed with plasmids. And then, you could at least separate this one out. So plasmids with an uninterrupted LACZ gene turn their bacteria blue. So uninterrupted is the one without the cut up recombinant plasmid. And then in the recombinant plasmids, the inserted gene interrupts the LACZ gene, and the bacteria remain their original color. So then, this one right here won't change blue. Bacteria which did not take up any plasmids also remain uncolored. So this one's uncolored, and this is the transformed bacteria that's uncolored. But then the one with the unchanged plasmid is transformed to blue. Yes, fascinating stuff. So now let's continue further. So now let's look at 9. So antibiotics are added. And these ones kill the bacteria. So what we have is because the plasmid contains the genes for antibiotic resistance. So remember right here. So we had this recombinant setup over here, or the plasmid itself had an antibiotic resistant gene over there, as well as some multiple cloning sites and LAZ gene for the blue color. So going back here. And now this is the recombinant plasmid. So we have antibiotic resistance genes right here. And we have interrupted this one's right here. This is the antibiotic. So this right here. That's the antibiotic resistant gene in this recombinant plasmid. Then what we'll have here is interrupted LAZ genes. Interrupted because you're adding extra stuff inside. And then the gene of interest that you've added is this red one. So that's this red one that you've added. That's going to be right here, gene of interest that you're adding. And it has the antibiotic resistant gene. So now you add the antibiotics, and then because the plasmid contains the genes for antibiotic resistance, only bacteria which took up the plasmid survive. So that means all the ones with untransformed bacteria, these die out. So all you're left with is blue for the unchanged plasmids, and then not blue for the transformed. So now you have, for the recombinant one. So now you have a way to tell which ones are the actual recombinant bacteria. So you'll have surviving bacteria. You'll have this one right here, the unchanged color, but with the recombinant plasmid. Then you have the regular plasmid that's blue. So now you know that these are the ones that you did. So these are surviving ones, and then dead bacteria don't have the plasmids at all. So now you have a way of separating them. And again, the plasmid of both have the antibiotic resistance genes inside. Now the next step is 10. The bacteria can then be sorted by color, isolating the bacteria which took up a plasmid containing the gene of interest. Yeah, so now you have this right here, bacteria containing unchanged plasmids in blue, right there. And then you have the not blue, the surviving ones, and the other surviving ones, but that have the plasmid containing gene of interest. So you have the recombinant plasmid and bacteria containing recombinant plasmids. And then what you'll have is you'll take out the color ones out, and then the uncolored bacteria can then be allowed to reproduce. So now you're going to reproduce these ones, and they're going to be duplicate, etc., more and more of them. So this is a diagram of molecular cloning using bacteria and plasmids and adding genes of interest inside. So absolutely fascinating stuff. Let's just zoom out now. Let's see how far we can zoom out. One more, I think. Okay, this would be good. So now we're out of there. And let's go back up here. So we had the DNA ligase. Yeah, I've been calling it ligase, but Google apparently calls it ligase. But ligase. Okay, so that's an interesting pronunciation. But I think ligase might be it. Yeah, so let's read up on that. So DNA ligase or ligase is a specific enzyme. A ligase or ligase. I'm saying both just because I think that the actual pronunciation is ligase, but Google pronunciation says ligase. So that facilitates the joining of DNA strands together by catalyzing the formation of a phosphodiester bond. Yes, so we go over that one. Yeah, all right. So the next one is genomic library right here, and this is if we scroll all the way back up before this molecular cloning. Remember we mentioned over here. So we mentioned molecular cloning as a method of genetic recombination, and other tools include using the use of genomic libraries, DNA microarrays, expression vectors, synthetic genomics, and CRISPR gene editing. So let's discuss these. All right, so let's keep scrolling. So a genomic library is a collection of the total genomic DNA from a single organism. Yeah, so a DNA microarray, also commonly known as DNA chip or biochip, is a collection of microscopic DNA spots attached to a solid surface. Scientists use DNA microarrays to measure the expression level of large numbers of genes simultaneously, or the genotype, or to genotype multiple regions of a genome. And so here is the video right here. I'll go over this video soon. Basically, what you have is some DNA sequences attached to a solid surface here, and then what you'll have is then you try to match it up, and then you expose it to free-floating other DNA sequences, and if they match up, and then they would either shine a color, and you can detect that it matched up with this known sequence. So this unknown becomes known once it matches up with this. So video, how to use a microarray for genotyping. The video shows the process of extracting genotypes from a human spit sample using microarrays. Genotyping is a major use of DNA microarrays, but with some modifications they can also be used for other purposes such as measurement of gene expression and epigenetic markers. In biology, epigenetics is a study of heritable phenotype changes that do not involve alterations in the DNA sequence. And before I get to the video, so genotyping is a process of determining differences in the genetic makeup, hence genotype of an individual, by examining the individual's DNA sequence using biological assays and comparing it to another individual sequence or reference sequence. Traditionally, genotyping is the use of DNA sequences to define biological populations by use of molecular tools. And it does not usually involve defining the genes of an individual. Yes, so it's used to see the differences in genetic makeup there, between an individual and a specific sequence that you're referencing, or another person's individual sequence, etc. And epigenetics is the study of heritable phenotype changes, or the observable traits that do not involve alterations in the DNA sequence. We'll get to this more later in this video. So let's watch this little video here. So if you click it, it goes to this DNA microarray Wikipedia page, and then if you could just click this right here. All right, so here I just made it bigger, and let's just play this right here.
A DNA reading starts with a plastic tube with your spit. In your spit, there are cells with your DNA. The spit is processed, so there is only DNA left. This is called DNA extraction. Your DNA is of course very long, but in addition to the extraction, it is cut up in smaller bits. All your DNA is then placed on a so-called microarray, and this microarray is inserted into a reader. A microarray has short pieces of DNA attached to it already. They're called probes. If your DNA string matches a probe, the two will bind together very strongly. Other probes may not match your DNA. Using fluorescence, one can have the bound DNA emit light into the reader. There are many variations on how to do that exactly, but generally it means that we can have each light correspond to a specific genotype. The light can then be read with a high-resolution digital camera And this way your spit sample can be translated into a list of what genotypes you have.
Mm-hmm. Yeah, so that's pretty fascinating stuff there. Alright, so yeah, let's get back to it. So basically you have these probes, these DNA sequences connected to the solid surface. Yeah, then you have the DNA that's extracted from a sample such as human spit, and then it's broken up. It's broken up into smaller pieces, and then if it connects to here, it shines a light. Then you have a high resolution camera that would spot all of the individual lights, and then basically encode them into a giant string of text. Yeah, corresponding to the genotype of each specific known sequencer, if it's on or off, etc. You have a giant long string of text. Yeah, which is quite fascinating stuff. So let's continue further. So we went over genotyping. So this is genotyping is biological. It uses a biological assay and comparing it to another individual sequence, etc. And a bioassay or biological assay is an analytical method to determine the concentration or potency of a substance by its effect on living animals or plants in vivo, or on living cells or tissues in vitro. Let's put a space there. Yeah, and vitro is in the lab, and then in vivo is in the organism itself. And in this case, the bioassay is seeing basically how much of the genotype is being expressed or is present in that sample. Alright, so now next setup is the expression vector, and we've mentioned it several times. So basically when we have the plasmid as a vector, it's basically used to deliver genetic information to other organisms. And yeah, so we have here other tools include use of genomic libraries, DNA microarrays, and expression vectors, and then we'll go over synthetic genomics and CRISPR gene editing. So expression vectors. Let's keep going to it. All right. So an expression vector, otherwise known as an expression construct, is usually a plasmid or virus designed for gene expression in cells. The vector is used to introduce a specific gene into a target cell and can commandeer the cell's mechanism for protein synthesis to produce the protein encoded by the gene. Yeah, expression vectors are the basic tools in biotechnology for the production of protein. So yes, so expression vector, it's like that plasmid vector, but in this case, it's designed to express a specific protein. Yeah, by inserting a specific gene into a target cell, and then you basically control the cell's mechanism for protein synthesis, which is very, very interesting. So you have a plasmid or a virus. And so note, vaccines are supposedly weakened virus and other similar setups like that into humans. So so technically it's a type of just expression vector, basically taking control of the cell's mechanism for protein synthesis to produce a specific protein. So you're essentially modifying humans, which is a quite interesting topic. So anyways, continue further. So vectors are also components of the current, I just noted that actually. So vectors are also components of the current COVID-19 vaccines, using mRNA vectors to get the body to produce a supposed, quote, coronavirus spike protein. Now that's an interesting topic to go over in detail in later video. So synthetic genomics is a nascent or new field of synthetic biology that uses aspects of genetic modification on pre-existing life forms or artificial gene synthesis to create new DNA or entire life forms. Yes, synthetic genomics is unlike genetic modification in the sense that it does not use naturally occurring genes in its life forms. It may make use of custom-designed base pair series, though in a more expanded and presently unrealized sense, synthetic genomics could utilize genetic codes that are not composed of the two base pairs of DNA that are currently used by life. So now we're getting into some next level science fiction type genetic modification. Yeah, so basically synthetic genomics uses non-natural sequences, and even possibly in the future or in classified tech, uses non-two base pairs setup. So you have 3 base pairs or 4. Yeah, to create a robo-human or a half man, half lion or something. So anyway, synthetic biology or synbio is a multidisciplinary area of research that seeks to create new biological parts, devices, and systems, or to redesign systems that are already found in nature. And so going further, so artificial gene synthesis or gene synthesis refers to a group of methods that are used in synthetic biology to construct and assemble genes from nucleotides de novo. We'll get to that in a bit. So unlike DNA synthesis in living cells, artificial gene synthesis does not require template DNA, allowing virtually any DNA sequence to be synthesized in a laboratory. And de novo synthesis refers to the synthesis of complex molecules from simple molecules such as sugars or amino acids, as opposed to recycling after partial degradation. Yeah, so basically you're assembling genes from nucleotides de novo, just straight up from the simple molecule sugars, amino acids, etc. Yeah, instead of recycling previous nucleotides that are partially degraded. So de novo is a Latin phrase, literally translating to, quote, from the new, but implying, quote, a new, quote, from scratch or from the beginning. Basically, creating nucleotides from the beginning or from the basic building blocks. Continue further. Going back to this, so the nucleotides de novo, so you're basically creating it from the phosphates and the ribose or deoxyribose, etc. Yeah, deoxyribose for DNA. So you're setting those up directly from those molecules. Yeah, as well as the nitrogenous bases, the nucleobases. Let's continue further. So genetic engineering, also called genetic modification or genetic manipulation, is a direct manipulation of an organism's genes using biotechnology. And so let's look at CRISPR now. CRISPR, or CRISPR, which is an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, is a family of DNA sequences found in the genomes of prokaryotic organisms such as bacteria and archaea. These sequences are derived from DNA fragments of bacteriophages that had previously infected the prokaryote. They are used to detect and destroy DNA from similar bacteriophages during subsequent infections. Yeah, so this is an interesting setup here. So CRISPR, remember, but it's usually everyone just thinks of CRISPR as that CRISPR genetic modification technology, which we'll discuss soon. But CRISPR, it stems from this basically repeating set of DNA sequences in the genomes of these single-cell organisms or prokaryotes of bacteria and archaea. So these ones are derived from DNA fragments of bacteriophages that had previously infected the prokaryote, and then these sequences are used to detect and destroy DNA from similar bacteriophages during subsequent infections. So they're derived from these bacteriophages after quote infection, and then a repeat encounter with a bacteriophage, they get destroyed. They get destroyed, or that DNA they destroy the DNA of the bacteriophages, of the new ones coming in. So going over here, so a palindrome, a palindrome is a number, is a word, number, phrase, or other sequence of characters which reads the same backwards as forwards, such as madam is M-A-D, and then M-A-D. It doesn't matter which way you go. Or race car. So if you go race car and you go backward, R-A-C-E-C-A-R, or this forward, R-A-C-E-C-A-R. So race car, same forward and backward. So hence these sequences are the same forward and backward. Going further, so a bacteriophage is also known informally as a phage, is a virus that infects and replicates within bacteria and archaea. Yeah, and here is just an illustration of the bacteriophage. Here's a structural model at atomic resolution of bacteriophage T4. And this is the scale 300 angstrom. Yeah, this is 300 angstrom. It's basically the A right here with a circle on top equals angstrom, and it's 10 to the negative 10 meters, or in other words, 10 billionth of a meter. Yeah, whereas a nanometer is a billionth of a meter, so basically 10 times smaller than a nanometer. Yeah, so here is how it looks like at basically the atomic level there of you can see individual atoms of each individual molecule, etc. So it looks like a little spider. So now here, so that's structural model, and now here's the electron micrograph. It looks like this. So bacteriophage P22, a member of the Podoviridae, or Podoviridae, by morphology due to its short, member of it, due to its short non-contractile tail. So basically, different setups of the images of the electron micrograph. You can split them up into different groups or families of characterization. So you look at it over here. So basically, you get this blurry little blob, but it looks like a hexagon there. So very interesting shape there. And there's these little little legs, and then this one has a short tail, non-retractable tail. This one it looks like it's a longer one. So then T4, this is a P22. Very interesting. And here's another image here next to a cell, bacteria cells, to make this a bit smaller. So in this electron micrograph of bacteriophages attached to a bacterial cell, the viruses are the size and shape of coliphage T1. So other types of other types of bacteriophages. Yeah, so basically, look at them like that. Supposedly, they're attached to this cell, right? There's a bacteria cell. It's very interesting setup there. Yeah, so we're still going to have to discuss the CRISPR technology. So basically, keep that in mind. So you have these bacteriophages. They supposedly infect bacteria. And then, infect bacteria or archaea, is basically single-celled organisms. And then after that initial infection, the sequences, there's sequences derived from those DNA fragments of bacteriophages. Yeah, from the remains. And then what you have is, basically they're going to be clustered, regularly interspaced, short, and palindromic. So they go, repeats basically same forward and backward. And then once those bacteria have already been pre-infected, then the next time they get infected, they detect and destroy DNA from similar bacteriophages during subsequent infection. So they're able to destroy the DNA in these bacteriophages that come and attack again. So keep that in mind. That's going to be utilized for CRISPR tech. So Cas9 or Cas9 stands for CRISPR associated protein 9. Formerly called Cas5, CSN1, or CSX12, is a protein which plays a vital role in the immunological defense of certain bacteria against DNA viruses and plasmids. And this is an interesting setup here. So note that plasmids and DNA viruses have basically can have a similar role. So, and in defense of certain bacteria against these DNA viruses and plasmids. Again, plasmids are just free-floating, circular loops of DNA, and is heavily utilized in genetic engineering applications. Its main function is to cut DNA and thereby alter a cell's genome. It is an enzyme that uses CRISPR sequences as a guide to recognize and cleave, cut so as to separate specific strands of DNA that are complementary to the CRISPR sequence, which is very interesting. So this Cas9, it's again used heavily in genetic engineering, and it's able to cut DNA, and then so it's able to cut DNA, and it's an enzyme that uses CRISPR sequences as a guide, so it can recognize and cleave specific strands of DNA that are complementary to the CRISPR sequence. So it can cut DNA to match up with this CRISPR sequence. And then Cas9 enzymes, together with CRISPR sequences, form the basis of a technology known as CRISPR-Cas9, or Cas9, that can be used to edit genes within organisms. Yes, this is all coming together now. So CRISPR gene editing, pronounced, quote, CRISPR, is a genetic engineering technique by which the genomes of living organisms may be modified. It is based on a simplified version of the bacterial CRISPR-Cas9 antiviral defense system. By delivering the Cas9 nuclease, again another enzyme, complex with a synthetic guide RNA or gRNA into a cell, the cell's genome can be cut at a desired location, allowing existing genes to be removed and or new ones added in vivo or in living organisms. Yeah, so very interesting. It basically uses synthetic guide RNA and plus this enzyme which is the Cas9 enzyme, that can cut DNA that can cut DNA using CRISPR sequences as a guide. So very fascinating. And now you're using that setup, but a simplified one with a guide RNA. So you can basically cut any desired or many desired locations, and then you can allow existing genes to be removed and or new ones to be added. So a nuclease. So this is a Yeah, this is dealing with this one, Cas9 nuclease. So a nuclease is an enzyme capable of cleaving or cutting and then so as to separate the phosphodiester bonds between nucleotides of a nucleic acid. So it's a nuclease. A phosphodiester bond occurs when exactly two of the hydroxyl groups, so we already went over this earlier, but just go over it, so which is OH, in phosphoric acid, H3PO4, react with hydroxyl groups of other molecules to form two esters, and OH is replaced by O-R bonds. And it's essentially it's that bond between the backbone of the sugar and phosphate groups in the nucleotide backbone, and then basically you're able to separate that with the nuclease enzyme that's capable of cleaving the phosphodiester bond between nucleotides of nucleic acids. Going further, so a guide RNA or gRNA is a piece of RNAs RNA that functions as guides for RNA- or DNA- targeting enzymes, which they form complexes with. Yeah, so basically you can use that as a guide. So you connect that RNA as guide to enzymes that you want to target other DNA or RNA, and then it makes a complex, and then it can link up to other setups. And now you're basically bringing in the enzyme to that specific target RNA or DNA. Yeah, and this type of complex here, so you have RNA and an enzyme, or protein. What we'll call this is basically it's a protein ligand complex. Is a complex of a protein bound with a ligand. Yeah, that is formed following molecular recognition between proteins that interact with each other or with various other molecules. And remember, ligand is just basically considered as a lump of different types of molecules. Yeah, that's connected to a central one. So basically, the ligands, let's just review that again. Yeah, so let's just search up ligand right here. Oops. So search up ligand. And right here. So let's go back here. So a ligand is a substance that forms a complex with a biological molecule to serve a biological purpose. The etymology stems from Latin ligare, which means to bind. And the complex is basically consists of a central atom or ion, and called the coordination center right here, and then the surrounding array of bound molecules or ions that are in turn known as ligands. So basically, you have complexing agents or ligands connected to a central atom or ion. So that is that. Let's continue back over here. So there's a protein ligand complex. Yeah, so basically a protein bound with a ligand, which is centered around an atom or ion. Alright, so we have that set up, and now here's the CRISPR-Cas9. Yeah, this diagram goes over the general overview of it. So let's say you have, right here, you have dsDNA, or double-stranded DNA. So you have the 5' over here, and it goes to the 3'. And then you have the anti-parallel, so the 5' all the way to the 3. So now you have your Cas9 setup right here. Yeah, so this Cas9, let's go up to it again. So the Cas9 is, it's the CRISPR-associated protein 9, and what it does is, its main function is to cut DNA and then thereby alter the cell's genome, and uses CRISPR sequences as a guide to recognize and to cleave specific strands that are complementary to the CRISPR sequence. And basically, it's an enzyme, that uses CRISPR sequences. So we'll continue further. So we have this Cas9 protein, or enzyme, and we have this guide protein, and this goes from the 3' it loops around all the way to the 5' like that. And this forms a complex with this. It's a protein ligand complex. So you have the protein, and then you have this ligand that surrounds an atom, and this ligand in this case is the RNA guide protein. And let's see what it does again. This functions as guides for RNA or DNA targeting enzymes, which they form complexes with. Yeah, so this protein is targeting a specific DNA sequence over here. And it uses this guide RNA. So now this guide RNA has a specific sequence at the end where the 5' goes this way. It's anti-parallel to the 3' like this. And now there is a specific cut zone right there. And there's a sequence right here, NGG or PAM, where this G and G is a nucleobase, and this N is any nucleobase. Yeah, and then followed by 2 GGs, or guanine. All right, so there's a basic general overview, and there's a target sequence, and then there's the cut via this Cas9 protein. Let's continue further. So this PAM, so a protospacer adjacent motif, is a 2 to 6 pair base pair DNA sequence immediately following the DNA sequence targeted by the Cas9 nuclease. Yeah, in the CRISPR bacterial adaptive immune system. This Cas9 nuclease, again, remember the nuclease is this enzyme capable of cutting the phosphodiester bonds between nucleotides. So it's able to cut these bonds. Yeah, connecting to the basically the backbone of the nucleic acid strand there. Yeah, and this PAM is a sequence immediately following the DNA sequence. So you have it over here. This is the one that's targeted, and it's immediately following that. And the canonical or typical PAM is a sequence 5' and then going from the 5' to 3', and then NGG 3', where N is any nucleobase followed by 2 guanines. Yeah, so 2 guanine or G nucleobases. So you have this NGG. So N is any nucleobase and GG, and that's a typical or canonical PAM setup there. And again, note the 5 and 3 ends refer to the directionality. This is the I believe the dash is on the outside. So the 5' and 3' refer to the directionality of a strand of nucleic acids, and it's about the backbones set up here. So there's nucleobases. This one is for hydrogen bonding, like that, to the other base, like that. And then this setup is here, so you have the 5' and 3 and all the way to the 3 like that, and this is where the backbones are connected, like that. And usually, yeah, we'll get to that in a bit. And then upstream would be the 5 one, so it's actually going down. So then if you give a direction to it, so the 5 is upstream, and then the 3' is downstream, right, like that. And these are where the other phosphodiester bonds are connected over and set up to the other setups. All right, so that's what we have. So the this image right here is a furanose or sugar ring molecule with carbon atoms labeled using standard notation. And then you have the 5 is upstream, and the 3 is downstream, and DNA and RNAs are synthesized in the 5 to 3 direction. So it's created from the 5 to 3. In this case, it'd be synthesized from there to there, or from this opposite to this side, like that. So you go from that end there, like that. Yeah, and this sugar ring, this one is the sugar ring is a furanose in general, but this is a ribose, has that H over there, so a type of furanose or sugar ring. And let's continue further. Let's look at this directionality again. So in the DNA segment shown, the 5 to 3 directions are down the left strand and up the right strand. So there's that bond right here. So there's the sugar, the ring, and then the phosphate backbone. And then you have the 5' phosphate deoxyribose backbones for DNA. You have AT, and there's the nucleobase hydrogen pairs. Hydrogen bonding connecting those over there. And this is downstream this way, so the 5' is going downstream, and then this up is on the 5 to 3 on the anti-parallel setup. That's fascinating stuff. Let's continue further. Yeah, so many of these tools have wide applications, such as creating medically useful proteins, or improving plant cultivation, which is basically preparing and growing plants, and animal husbandry, which is animals raised for food. Animals raised for food and other products. And human insulin, for example, was the first medicine to be made using recombinant DNA technology. So that's very interesting. Other approaches such as farming. This is farming with a PH instead of a F, can produce large quantities of medically useful products through the use of genetically modified organisms. So MES note, farming with a PH, which is a portmanteau, which is a blend of words of farming and pharmaceutical, refer to the use of genetic engineering to insert genes that code for useful pharmaceuticals into host animals or plants that would otherwise not express those genes, thus creating a genetically modified organism, GMO. Farming is also known as molecular farming, molecular farming with a PH, or biofarming, again with a PH. And a medication right here, referring to as pharmaceutical. So a medication also referred to as a medicament, or a medicament or medicament, or medicine or pharmaceutical, or pharmaceutical drug, medicinal. Or a medicinal drug, or simply drug, basically just different variations of the same word, pharmaceutical. Drug is a drug used to diagnose, cure, treat, or prevent disease. And a drug is any chemical substance that causes a change in an organism's physiology or psychology when consumed. Yeah, so going back to this farming, the pH. Yeah, basically you're inserting genes into animals, to produce, so then have them produce specific substances that you could use for drugs. Yeah, so it basically insert them that they wouldn't naturally or otherwise have in those organisms, and then you would produce specific quantities of useful products or substances that you could utilize for them, which is quite fascinating.